Good day to the viewers. In this video today we are going to discuss about Krebs cycle. Let's see about the introduction. Krebs cycle is also known as tricarboxylic acid cycle or citric acid cycle because citric acid is the first product of the cycle which contains three carboxyl groups. Citric acid is also known as citrate when it is ionized. It is a sequence of chemical reactions that use oxygen as part of cellular respiration in all living cells. In eukaryotes, the Krebs cycle occurs in the mitochondria, but in prokaryotes, it occurs in the cytoplasm. Each cycle yields one molecule of ATP, three NADH molecules, and one molecule of FADH2, and these are required to synthesize ATP for cellular respiration activities. The citric acid cycle is the final common pathway for the oxidation of all biomolecules like proteins, fatty acids, carbohydrates, and amino acids. Molecules from the other cycles and pathways enter this cycle through acetyl-CoA. This cycle is also particularly important as it provides electrons, high energy molecules to the electron transport chain for the production of ATP and water. TCA cycle was proposed by Sir Hans Krebs, a German biochemist in 1937. He was awarded the Nobel Prize in 1953 for his contribution. Now we'll see the various steps involved in TCA cycle, the preparatory step which begins with pyruvate. Pyruvate cannot enter the Krebs cycle directly. In a preparatory step, it must lose one molecule of carbon dioxide and become a 2-carbon compound. This process is called decarboxylation. The 2-carbon compound called acetyl group attaches to the coenzyme A through a high energy bond. The resulting complex is known as acetyl coenzyme or acetyl-CoA. During this reaction, pyruvic acid is also oxidized and NAD plus is reduced to NADH by pyruvate dehydrogenase complex. This multi-enzyme complex is responsible for the conversion of pyruvate to acetyl-CoA. Therefore, pyruvate dehydrogenase complex contribute to linking the glycolysis pathway to the citric acid pathway. The first step is formation of citric acid. The 4-carbon oxaloacetate condenses with 2-carbon acetyl-CoA to form 6-carbon compound called citrate. The enzyme is citrate synthase. The hydrolysis of the thioester bond in acetyl-CoA drives the reaction forward. This reaction is an irreversible step. The second step is formation of isocitrate. Citrate is isomerized to isocitrate by acotinase. This reaction is a two-step process. At first, one water molecule is removed from citrate, forming cis-aconitate. It is a transient compound with a very short half-life. Immediately, one water molecule is added to aconitate to form isocitrate. Thus, the position of hydroxyl group is shifted. Third step is formation of alpha ketoglutarate. This reaction is a two-step process, both catalyzed by the same enzyme, isocitrate dehydrogenase. In the first part of the reaction, isocitrate is dehydrogenated to form oxalosuccinate. It is an unstable compound which undergoes spontaneous decarboxylation to form a alpha ketoglutarate. And the NADH is generated in this step, later oxidized in electron transport chain to generate ATPs. Isocitrate, it is a 6-carbon, undergoes oxidative decarboxylation to form alpha ketoglutarate, which is a 5-carbon compound. In this reaction, one molecule of carbon dioxide is liberated. The isocitrate dehydrogenase as isoenzymes, the mitochondrial form utilizes NAD+, while cytoplasmic enzyme is NADP+, is independent. Fourth step is formation of succinyl-CoA. Alpha ketoglutarate is oxidatively decarboxylated to form succinyl-CoA by the enzyme alpha ketoglutarate dehydrogenase. The NADH thus generated enters into electron transport chain to generate ATPs. Another molecule of carbon dioxide is removed in this step. This is the irreversible step in the whole reaction cycle. The enzyme alpha ketoglutarate dehydrogenase is a multi-enzyme complex having 3 enzyme proteins and 5 coenzymes. This is similar to pyruvate dehydrogenase reaction. The first two enzyme activates are similar to the corresponding components of pyruvate dehydrogenase complex, and the third enzyme is the same in both complexes. In the fifth step, generation of succinate. The reaction involves a substrate level phosphorylation whereby a high energy phosphate is generated from the energy trapped in the thioester bond of succinyl-CoA. The enzyme is succinate thiokinase. A molecule of GDP is phosphorylated to GTP and succinate is formed. The GTP can be converted to ATP by reacting with an ADP molecule. And sixth step is formation of fumarate. Succinate is dehydrogenated to fumarate and unsaturated dicarboxylic acid by succinate dehydrogenase. The hydrogen atoms are accepted by FAD molecules. The FADH2 then enters into electron transport chain to generate ATPs. The enzyme is a flavoprotein. The succinate dehydrogenase is competitively inhibited by malonate. And the seventh step is formation of malate. The formation of malate from fumarate is catalyzed by fumarase. The reaction involves the addition of water molecule. Only L-malate is formed. And the last step is regeneration of oxaloacetate. Finally, malate is oxidized to oxaloacetate by malate dehydrogenase. The coenzyme is NAD+. The NADH is generated in this step, which enters the electron transport chain when ATPs are produced. The oxaloacetate can further condense with another acetyl-CoA molecule and the cycle continues. Now let's see the ATP generation in Krebs cycle. As per the old calculation, we already know that one NADH molecule will produce 3 ATP molecules. So 3 NADH will produce 9 ATP molecules. Next, 1 GTP is produced, which is equivalent to 1 ATP, and 1 FADH will give rise to 2 ATP molecules. So totally 12 ATP molecules are generated in one cycle. As we know that one glucose molecule breaks down into 2 pyruvate molecules, which is eventually goes into 2 separate Krebs cycles. So on the whole, 24 ATP molecules are generated as per the old calculation. But according to the new calculation, 1 NADH will give rise to 2.5 ATP. So 3 NADH will produce 7.5 ATP molecules. 1 GTP, which is equal to 1 ATP, and 1 FADH2 molecule produce 1.5 ATP. So totally 10 ATP molecules are generated in one cycle. For 2 cycles, totally 20 ATP molecules are generated. Thank you for listening the video.